Okay, hey everyone. Hope you can see my slides. So yeah, first of all, thank you very much for this kind introduction and also thank you very much for having me here. So my name is Stefan Lengauer from the IVC, the Institute of Visual Computing, and today I have the honor to present to you one of our latest research topics called Droplets, which is a novel marker design for visualizing proportional relations, which also have some sort of spatial embedding. Yeah, we call them droplets to their characteristic shape, which you will see in a minute. Okay, yeah, the idea for this design originates from, as mentioned, the Biomed VUES challenge at IEEE VUES last year. So in general, a key challenge for cell biologists lies in the understanding of spatial patterns in tissue samples. And the key technique employed in this context is, as mentioned, called transcriptomics and refers to the simultaneous capturing of gene expressions as well as spatial coordinates. Yeah, depending on the technique, the resolution can vary from subcellular over single cells over spots, which represent multiple cells. And in the given challenge, we look at the latter case, namely that such spots represent multiple cells, which can also be superimposed to a corresponding hemadoxylin and eosine stain of a tissue sample. That's the orange-violet stuff you can see here. So for the challenge, the cell types which we use have been obtained with the so-called ST-deconvolve by Miller et al. And each spot, or pixel as we call them, consists of both the proportional relations describing the prevalence of different cell types, as well as a location in the spatial domain. Yeah, here in this example, they are represented by pie charts. But as you can instantly see, there are some shortcomings in this technique. So areas of coherent cell type composition are inherently hard to spot as there are many spatial disarrays between the different components. So with respect to this example, you can see that there is a very prominent reddish component in the center of the tissue sample, but that's about all you can see. While in the data, there are actually a lot more patterns which are latent in this visualization. Yeah, pie charts are just not very good in small multiples visualization. I think it was also quite fittingly described by Tufte and Griff Morris with the words, the only worse designed than a pie chart is several of them, for then the viewer is asked to compare quantities located in spatial disarray, both within and between pies. So yeah, so much on the objective of the challenge, which was basically to come up with a design which can improve upon this sort of standard visualization. And yeah, the way we approached this was to, first of all, make up our minds regarding what an optimal design should entail. So we agreed upon that the available space should be used as efficiently as possible so that the underlying image can still be perceived. Then we want to visually enhance regions of coherent cell type compositions, clusters, so to speak. And also, we want to visually reflect the association with local clusters, as there are no hard boundaries between such clusters, but rather smooth transition between areas of different compositions. Well, as a starting point, we looked at different baseline visualizations for proportional relations, such as bar charts, tree maps, etc. But it's quite obvious that they all suffer from the same shortcomings as pie charts. They're just not really appropriate for small multiple visualizations. It's really hard to spot any patterns here. Yeah, then of course we also looked into literature where we found quite interesting ideas. One closely related concept is, for example, this Sixtel idea by Mert et al., which uses a spider chart marker for multivariate data in also a small multiple visualization, and also overlaid over medical images. So quite interesting. And then we were also heavily inspired by this winglets analogy by Liu et al. That's not really proportional data, but scatter plots. But still, I found the idea quite intriguing with this winglets attached to the scatter points, which indicate sort of yeah, cluster borders. And that's something we also strive to achieve with our visualization. Right. So, oops. if we go back to this slide, I would say without much further ado, I will pitch our proposed idea. Okay, there is. So basically our idea is to use separated yet tightly packed shapes, one for each class, which reflect the cell type. Yeah, we decided for the most simple shape, a circle, so to speak, where the radius reflects the contribution of the class. But additionally, as you can see, we equipped the circle with a protruding tail, which points in the direction of the next accumulation of the same class. And also the manifestation of this tail, how pronounced it is, depends on how far off this accumulation is and how prevalent it is. So yeah, that's our design concept. And I would say let's have a closer look at how we actually define this design. Yeah, but first of all, we need to clarify some of the terminology which we will use throughout the next slides. So one particular spot or pie chart or whatever we call it, we now refer to as a pixel. So this is a pixel. And capital X is the index set of all of our ST pixels. Similarly, we have different classes. So we have a set of classes K. And in this pie chart, we have 5 different classes. The reddish one is K equals 0, the greenish one K equals 1, and so on and so forth. Then P of X is the center point of the pixel and is the spatial embedding of this pixel X. And also WXK is the class K's contribution to pixel X, so the proportional value. That's the floating point value in the range from 0 to 1. So, for example, WX0, the reddish component, has a contribution of 0.267-ish, roundabout. Okay. Oops. All right. So as said in our proposed design, each contribution W is visualized with such a droplet shape, which you can see here. And one specific droplet is completely defined by 4 parameters. So firstly, we have the radius of the base circle, then the eccentricity, which is like an ellipse, the ratio between the radius and the droplet's most extreme point, right at the tip. As I said, the droplets exhibit an adaptive eccentricity, so yeah, the eccentricity could become 0, meaning that it will become a circle in case if there are no nearby clusters or so. Then obviously we need an orientation to orient our droplet in the pixel. And last but not least, we have to have the center point, which is the location within the pixel. Now the big question is, how do we define all of these parameters? Yeah, for the radius, it's rather straightforward. The radius can be just mapped to the respective contribution W. But for the eccentricity and the orientation, on the other hand, we need some knowledge of both the strength and the direction of nearby clusters. So in other words, we need to have a potential describing the attraction to neighbors. Yeah, that's a vector field here given as psi of xk. Note that we have a different potential for each of the classes as they are considered independently. Yeah, the same potential is also used in the determination of the position of a droplet within a pixel as they try to strive towards their respective clusters, so in the direction of their tail. And also for the positioning, we need to consider the positions of all other classes of the same pixel to avoid collisions and such. Okay. Yeah, then regarding this attraction potential which you mentioned, it's actually quite simple. Our premise is that a droplet is attracted by all other droplets of the same class, but relative to their respective contributions as well as their distances. So the attraction potential at an arbitrary point P is just the product of the contribution weighting as well as yeah, multiplied with distance weighting summarized over all pixels we have. And yeah, we employ a distance weighting modeled over Gaussian kernel, so a fast decaying proximity kernel with a fixed variance. And the weight is proportional to the contribution of a pixel. So actually what we have is a Gaussian mixture model, which describes our potential. Yeah, we can also visualize this quite easily. I've generated such a toy example where we can quite clearly see this. This is a regular grid of locations where we have two classes only, which have a balanced contribution of 50-50 each. I hope you can see the small pies in the slides. So this is a homogeneous field, basically, only within a circle in the middle. We have an unbalanced contribution with a 9-to-1 ratio of the first class, the reddish component. Okay. And if we now plot the potentials for both classes, the reddish and the bluish class, it's something like that, which we can see here. So we can see that the potential for the red class kind of forms a high plateau in the circle area. So there is a large potential which funnels out at the border of the circle, and everywhere around at this homogeneous region, it's basically zero. And for the other class, for the bluish one, it's quite the opposite. There's a depression in the middle, forming a low plateau, so to speak. And yeah, there's an ascent when you go the direction outwards the circle. Right. So yeah, but that's a scalar field basically. But what we want to have are vectors describing us the direction of these potentials. So yeah, we need to transform this scalar field into a vector field. Luckily, that's quite easily to obtain by differentiating this potential. So we need to employ a partial differentiation. That's also not too complicated for these Gaussian kernels. It's just the, as you can see here, if you take this equation, it's just the proximity kernel as well. Again, multiplied with the, we need to apply the chain rule, so multiplied with the exponent of the kernel, which is this additional term right here. And we do that in x and y direction, our spatial dimensions independently. And we can again plot this, which you can see down here. And there is not much difference between both classes in terms of the quantities, but if we plot the circles, which give us the arrows, which give us the directivity, I hope you can see these small arrows here, then we can see that the red class droplets will try to strive towards the center of the circle at the border of the circle. And it's quite the opposite for the bluish class, which is in the minority within the circle. So they, yeah, one could say they try to get out. Right. Okay. Yeah, so much on the attraction potential. Yeah, with the attraction potential, the eccentricity as well as the orientation is now fully defined, and also the locations of the center points to some extent. Yeah, only for the center points, there's still a bit of a question mark because we need also to consider the locations of all other droplets. And that's unfortunately a bit more complicated in a first implementation, which we also submitted to the challenge. We employed a physical model to this end with different forces, making sure that droplets are attracting each other. Then we had a collision force, making sure that they're not overlapping. We had a gravitational force dragging the whole assembly towards the center of the pixel, and we had a small force which made sure that components try to strive the direction of their orientation. Yeah, I mean, it worked quite well, but the problem is it's quite sensitive towards this force balancing. It's also dependent on initial condition, and it's usually a bit slower than other solutions because you need to have a lot of iterations and so. So yeah, our solution to this end was to change this layouting to an algorithmic approach, which I will also show you quickly. It's actually quite a simple algorithm which we developed to this end for layouting the different droplets in a ST pixel. And it goes like this. So first of all, we initialize all droplets position p of x with the center point of the pixel. Quite trivial. And then we sort all the classes according to their contribution. So note that in many pixels, we don't have all of the classes present because some of the contributions are zero. Actually, it's quite often the case. So in most cases, we have just a fraction of the classes present. So K is the sorted set of the droplets. And for the trivial case that we have just a single class, we can just return these positions because it's quite all right if the only present component is sitting right at the center of the pixel. So we're already done. But usually we have more than one component. So we need to do some collision avoidance or uncolliding. So if you have more than one component, we take the second component. This is the smaller droplet here, which you can see. So it's pk1. And we move it in the direction of its respective potential field for an amount which amounts to the combined radii of both droplets. So that they are touching but not intersecting. And finally, we do a small readjustment in the direction of the center. Okay, so those works for two components. For every additional component class we want to add, we do the following. So we take the kth element, so the next biggest element from the classes we still need to consider, and we compute a set of candidate positions where we could place our marker. So our premise here was that we want to ensure tight packing, so we determine positions which touch at least two of the remaining or the other already present droplets. So in this case, this would be those two bluish circles. Yeah, and from this set, we discard positions which are invalid because they're intersecting with existing droplets. And right now, there are no intersections, so both positions are valid, but could be otherwise. And then from the candidate positions, we want to determine the optimal position. And to this end, we use a angle criterion, meaning that we look at the angles which the respective positions form with the angle of the potential field. So, oops. In this case, we can quite easily see that the angle with the candidate position at the lower half is much smaller than the other one. So obviously, this is the best placement for our third component. Right. And yeah, again, a small readjustment in the direction of the center. And that's basically what we do for all remaining classes until there are none left. So yeah, for our next one, we have 6 candidates. 3 of them are invalid. So this leaves these 3, and so on and so forth. I think you get the idea. Right, and yeah, that's basically all for the design of our marker and for the placements. Now we have everything for drawing these droplet markers, and with that we can now actually look at some of the results. So our most important or go-to result is from the challenge dataset, which you can see here. So this is, as I mentioned, an H&E diagram, H&E stain of tissue samples, and here we have quite a large number of components. So quite a large number of pixels, I mean. So we had almost 3,500 pixels and 9 different classes labeled from X1 to X9. And if we now look at the side-by-side comparison of our droplets at the bottom and the pie charts, which you can see here, yeah, we can see some quite significant differences. For example, the reddish component at the center here is much more pronounced. The borders are clearer. There are much more high-frequency variations which we can observe. There is, for example, this turquoise component surrounding, or this turquoise component surrounding all of the tissue sample. There is this very subtle violet streak at the top left part of the tissue, and yeah, other quite interesting patterns which we can observe as opposed to the pie charts. Yeah, here we have a close-up to this center area. This is, of course, subjective, but I would argue that our pie charts are much cleaner and calmer than our droplets are much cleaner and calmer than the respective pie charts. A lot more pleasing to look at, and I think you can see patterns better. Yeah, please note that the droplets here have almost no tails. Only here at the right-hand side, where we have some tiny contributions at the fringes of this big reddish cluster. That's because it's quite a homogeneous area. But if we look further down, here's a second close-up. If we look further down at this big reddish component or cluster, we can see here where the X1 cell type fizzles out into the neighboring tissue. We can see the small droplets trying to strive towards their respective cluster centers. And yeah, I would say that's quite an improvement um, over this pie chart, at least. Okay. Yeah, so much on first results. As you might have guessed, our droplets design was developed for a challenge data set, and hence it's quite custom to this one, but development continues on these droplets, and we're now looking forward to evaluate it on other use cases. So a couple of examples we have in mind are other medical related stuff like MRI images, as you can see an example here, but also socio-demographic properties with a geographical context is something we are looking into right now. So these are two examples of, yeah, which my colleague just generated hours ago, so I can't describe them that well, but they depict two metropolitan areas, Berlin at the top and London at the bottom. And I think the variables which are encoded here are residential and forest areas, the reddish and orange components. But that's just a penny for your thought. Yeah, but generally any data which combines proportional relations with spatial embeddings should be usable. So if you have any ideas for further use cases, please just let me know. Then from a technical perspective, we're also looking into further ways to improve efficiency. The previously shown examples were generated with a naive Python implementation, and they took a couple of seconds to generate for the tissue samples on commodity hardware, but we also want to further improve this efficiency. My colleague Peter Waldert, who is also here today, is also working on a TypeScript implementation for web embeddings. And for some use cases, we also want to overhaul our methodology to adapt it for non-regular grids, which is now a limitation because we have this static variance in our Gaussian kernels. But yeah, that's about it. And yet another idea is to modify the underlying image, like making it more transparent in homogeneous areas and vice versa. So these are just a few examples for future directions we're planning to go to. All right, and yeah, this brings me quite to the end of my talk, actually. To summarize, I think our design shows quite promising results for the given problem. We have both a Python and a TypeScript implementation. Here you have the respective links. The TypeScript implementation is not yet online, but should be there eventually. And a final shout out to our project, which funded this research, the EU Horizon Project Hereditary. And yeah, with that, I really want to conclude my talk. And thank you very much so far for the attention. And I think I forward to my other speaker.